Welcome back to Paleo Talks, everybody. We are on episode 60, and today we are with Juliet Brophy, joining us from Louisiana. How you doing, Juliet?
Hi, good, great, thanks.
Wonderful to have you, and so excited about this topic today. And before we get into that, let's go over to David and tell us a little bit more about the show.
Sure thing. For any newcomers, the way that Paleo Talks works is here in just a second. We're going to do an introduction, have our guests introduce themselves, and then launch into our presentation. Today, we're talking about hominids, which is a rare topic and an exciting topic for us. Most of the program is going to be the guest presentation, and then once that wraps up, the rest of it is audience Q&A. So our panel of people here will probably have some of their own questions, and at that point, we will also ask the audience to send in your questions for our guest. You can go ahead and put your questions when the time comes in the Facebook comments, and as always, if for any reason you can't put a comment on Facebook, you can message us at the Gray Fossil Site's Twitter or Instagram account with your question. I'll be keeping an eye out over there as well.
All right, thank you, David. David is our science communicator here and takes care of all the logistics and saved us today in getting things set up as we were having some technical difficulties.
Minor technical issues today.
And as usually, we have Chris Widga with us as well. How you doing, Chris?
I'm good. Just on the other side of the wall from you. So
I know, and your technologies appears to be working, and that saved us today as well. So that's awesome. We're coming to you from the Center of Excellence in Paleontology, which oversees the Gray Fossil Site as part of East Tennessee State University here in Johnson City, Tennessee. And again, this is episode 60, so quite a milestone for us. And it's great to have this hominin talk for that. And caves, South Africa, I just couldn't be more excited about that. And Juliet, what we usually do at this part of the show is just turn it over to you and let you introduce yourself to everybody. Tell them about your background and how you got into paleo in the first place and how you ended up doing work in South Africa and finally to LSU.
Okay, well, thank you very much for having me. I'm really excited to be here and talk about Homo Naledi and the new cave systems. I am a biological or paleoanthropologist, and my dad fancied himself an amateur archaeologist, so I was actually able to go on a field school dig right the summer before I went to university, and went down to excavate in Belize and had a great time. Chose my university kind of late. I ended up going to Michigan and kept taking anthropology classes, cultural and biological, and I literally fell in love with a fossil, a picture of a fossil that is just near and dear to my heart, Mrs. Blez. I keep her on my desk all the time. And I was like, I'm going to go down and study her, which is ironic because I focus more on teeth and she has no teeth, but I do love her. So I took a lot of those classes. I finally got onto school working as a TA with the Duke Field School. And so I finally got down to South Africa to do some studying. And there I met up with Daryl DeRuyter from Texas A&M. And so I started working with him. He kind of was my master's advisor, even though we weren't at the same university. I was at the University of Tennessee for my master's with Andy Kramer. And then I went to Texas A&M for my PhD. And so continued my work in South Africa. So I did all my master's and PhD work there. I got a job at Loyola University in Chicago, which is fantastic. But it only has undergrad options, and it was a short-term position. And so eventually I got the job. I actually applied for a skeletal biologist job down here at LSU, but it's been absolutely fantastic. Lots of research support, and been able to continue all my work here, work in South Africa.
That's fantastic. And when was the last time you got to go to South Africa?
2019, right before COVID.
I think I have Miss Blez somewhere behind me as well. So part of our same decorations around.
Yes, that's my personal Mrs. Blez, mind you.
Nice. All right, well, let's go ahead and get started with your presentation.
Okay, great. Oops. Share screen first.
Sounds like we have several different areas of overlap. You've been in Tennessee, I worked in South Africa, and I've worked in Belize as well.
Oh yeah, wow. Okay, how's that look?
That looks great.
Fantastic. Okay, so title of my talk is Homo Naledi, a bite out of history, because I do specialize on fossil teeth, but I'm going to talk about the overall discovery of the site, the context, and then the newest information, a little bit about the implications, depending on what time frame that we have here. So the site was originally, let's go here. So the site is located in the cradle of humankind. It's a blow up of what that area looked like. And so just to remind you some of our most famous specimens from there. So this is the Tong child or Taung child. There's Mrs. Plez, of course, I had to include her in here. And so she was found at the site of Sturkfontein right there. We've got Australopithecus robustus that was found. This one was found at Svartkrans over here. And then another specimen, Australopithecus sediba, found in Malapa. So this whole cradle humankind area has a lot of ancestors found at the site. And so here's where Rising Star is located down here, the Rising Star cave system. So it was actually found by some recreational cavers, came across some bones and contacted Lee Berger at the University of Witwatersrand and said, hey, we found some specimens here. So he went out with some of the cavers from the university and found more of the bones. They found the original chamber that they were looking at, and he realized there was a lot of fossils to be found here. He put out a call on Facebook for literally small-bodied hominins to come and excavate the site. And he had, lots of applications. It was a little bit I don't think it was as many as he had kind of hoped, but you had to drop everything and go down there in the fall. So it's an amazing opportunity. Not so much for people in academia that couldn't drop everything. So he ended up having 5 women and 1 male, and at the last minute, the male dropped out. So it's 6 female cavers, and who later on the media dubbed the underground astronauts. So here's some of the cavers, to some of the local cavers, as well as the 6 that came down. But you can see the really hard to reach remote passages that that was found that were in order to excavate the material. I did want to show this picture up here. This is my picture. So directly beneath this is the first chamber where the original bones were found, the delated chamber. Not much to see, but that's, again, if you're going directly down. But from the opening cave to about where that chamber is about a length of a football field. So they excavated in the fall of 2013 for about 2 weeks, and another couple weeks, 2-3 weeks in the spring of 2014. And they had so many fossils that were coming out of there. This is just a caricature of what that would look like of the Dinilated chamber, the first chamber. So we excavated just the top layer here, right? And which was somewhat difficult, again, with all the provenience to try to get great provenience down within the cave. But we have a 3D printout of what this whole top layer would look like before they were excavated. But then they found that the concentration was just really dense, and some of the breakages were a little bit fresh, so they had to switch to barefoot excavations. And then there was one area where they found some long bone areas, and they just didn't want to break them to get broken. So they started excavating in this smallish area here, about the size of a large coffee table, took that down a little bit. And again, after only about 6 weeks, 5-6 weeks of excavation, came up with just over 1,500 specimens. So like, okay, we're going to stop for a little bit and see what we have. And this is an example of some of the specimens that were found. I mean, this is an articulated foot. So this is part of what this came out to be. So we have them, again, all the numbers associated right next to each other for the provenience. The other thing you can see in this picture is that in some spots, there's literally more bone than sediment. The only sediment that we're getting from here is coming from the cave walls. So again, the concentration is quite high. So here we're looking at part of the foot and then the lower limbs. Again, totally in articulation. We don't have this for every specimen, but again, there's a lot of pieces that we know are directly associated with each other. So again, after about 6 weeks of excavation, over 1,500, 1,550, and that was just when they basically stopped. There was still more that needed to be processed. So from that, Lee Berger put out a call again on Facebook for early career scientists and recent PhDs that had data to help identify this specimen, to be the primary analysts of these specimens. This is pretty unprecedented in paleoanthropology. This is not something that's typically done. And so it was a very awesome opportunity. He had about 300 applications, and he ended up with a team of about 30 individuals, and that consisted of from about, I think, 12 different nations, 12 different countries were included, as well as some senior scientists that were there as well to kind of help that had been involved as primary analysts on other sites as well. This is not a complete picture, but this is most of the group that went down. So this was part of what's called the Rising Star Workshop. And we went down there for about 6 weeks in 2014. I'm getting my years, I kept thinking it was 2015, but 2014, we went down there for about 6 weeks and intensely studied these fossils every day. And then at night, we would sit around and chit chat about them, what we found. And we were divided up into our little group specialties. So I worked in the tooth booth, but there was the hand area, handland, and hip heaven. People were, again, divided up in their different specialties. And so, again, it worked concentratedly for a really long time, for every day during those 6 weeks, and then continued to work on anything where we didn't need the fossils right next to us at night for those 6 weeks. So dissected, analyzed every bit of information we could squeeze out of these fossils. And after the 6-week period, we determined we did think that we had a new species. And it was based on the suite of characteristics that we found throughout the specimens, not any particular piece. Because one of the things that I'll focus on is the mix of characters that we have within this overall specimen. So that's what I'm going to do, just go an overall of what fossil specimens have been found and what they can tell us, what the morphology look like. So this is dentalated hominid 1, and so we've got part of the cranium, but we have 6 individuals based on the cranial pieces. So we've got this back area, the base part of the basicranium, and then we've got a part of the maxilla and mandible. So this is what we typed as the holotype specimen. But there's 6 different pieces, and we put these 2 with each other. We can help to reconstruct what the overall skull would look like. I really like this picture because one of the great things about Homo naledi, one of the things that helped put this specimen into the genus Homo, is overall the shape of the skull. So it's got this more globular shape to the skull versus those long, low vaults that we see in other species, such as Homo erectus or even the Australopithecines. So we start to see the early signs of a forehead, the outline, and again, I like this picture because it's got the outline of a modern human here. Modern humans have a very tall forehead, somewhat weird, very recent, very tall back region of the skull, the occiput region, and then tall side parts. And so we don't see that as much in other species, these really high areas to the skull. So while it has the look of a modern human, a more derived look or modern-like shape to the skull, it is very small. So the cranial capacity comes in at about 465 for the females and about 560 for males. So I'll put this in comparison, the average modern human size is about 1,400. So it's significantly smaller.
And what size would the chimpanzee be, Juliet?
Chimpanzee would be at the lower range, within a 440, within the 400 range, would be a large male. So this is one of those parts of that reconstruction that we had. So we had part of this cranial here, and then we can mirror image that, use that to create this piece of the skull. And then one of the women on our team, Heather Garvin, looked at cranial capacities, reconstructing cranial capacities, and so that she was able to put together this image. So again, it was really great that we had this really awesome team to it. So here's some of the dentition included in that dentalated hominid 1 that we looked at. I think arguably, obviously, the dentition is the coolest part of the site. There's so much we can learn about it. And so on all of our left, so we've got, these are some examples of the deciduous teeth. So in that first chamber, the dentalated chamber, we have 21 deciduous teeth, which is a lot to have at a hominin site. This is our, again, that dentalated hominid 1. So we have the complete mandible here, and then we have part of the maxilla up here. This is a mirror image. And then we have our grandpa specimen here. So we have really old specimens. So you can see that some of the enamel, the outer coating of the tooth has worn away. So we get into some of that dentition. So we have 190 whole or fragmentary teeth found at the site in the original dentalated analysis. And they were all different ages. So we had a minimum number of 3 juveniles and then some adults. And then again, we have this one really old individual. So again, a wide span of ages. So just a little bit more about the dentition. Again, it's the coolest. So we've got smaller anterior teeth than the Australopithecines that we see before, certainly within South Africa. So this is more Homo, more derived like us. We have some of the post-canines, so behind the canine teeth are generally more like early Homo in their shape. But we do have this interesting, what we call a molar size gradient. So in modern humans, the third molars are going to be the smallest. The second are typically smaller, and then the first are going to be the biggest. Sometimes 1 and 2 can be the same size, or that can change. But in general, the third molars are always the smallest. The primitive condition, more like the Australopithecines, like it was in the past, is that the molars get bigger as they go back. So while in general we have several Homo-like characteristics to the teeth, we have this primitive molar size gradient, where again, the first molar is the smallest. So some of the other characteristics that are unique and interesting, and mostly unique to Homo naledi, is that the canine here has this like mitten or cusp, this a little cuspule or cusplet, so it gives it this mitten shape. So we only see that in a couple other specimens. We have a couple specimens in Demnisi that have similar characteristic. And another one is the mandibular P3s. So this is something that I focus on. I looked at the occlusal crown shape, or the crown shape morphology of these fossils. And that's one of the things in that when I look at the shape of that tooth, it looks unique. That was one of my main contributions, is that it doesn't overlap with any other hominins that I could see in South Africa, East Africa, anybody, with anybody that I had. So I'll talk a little bit more about that here in just a second. This is a picture of the tooth booth. So I want to show you that, again, we had an amazing team. And in this booth is where we described and analyzed, again, try to get any bit of information that we could about these teeth. Everybody sans the baby. Esme was there. That's Matt Skinner's child. But she didn't contribute too much to the analysis, but she was in the picture. But again, this was a great, like I said, group effort. We put all our heads together, and this is not a complete picture. This is just who was there on picture day, I guess. But the other reason for telling you about this, other than highlighting what a great team that I worked with, was also that we were dividing these teeth up. Each of these sections are the rows. So this is how we try to determine how many individuals we had. So we looked at like tooth type, wear, age, sex, anything we can use to help determine what would go together, again, to try and determine the individuals. In the end, in the Naledi chamber, we think there are 15 individuals, and again, that's based on the dentition. The other thing that we would look at is that as you have a tooth in your mouth, as the second one comes up and occludes next to the other one, it's going to create this interproximal facet. And so it's only going to be unique to these two teeth. So we would try to look to see, okay, can we match the interproximal facets? Because we had so many articulated parts to the skeleton, we could do that with some of these specimens, and that was just awesome, I think. So that's another reason why I show this picture. And that's how we could do things like this, put together this individual. This is our best mandibular specimen, 1400, that's found in Naledi. We have the unoccluded, unworn first molar from the tooth germ, and then we have teeth from the other side. So we think this was one of the juvenile individuals, again, from both sides, and that's pretty cool. I've been focusing a lot more on the deciduous dentition of late, and Shar Bailey, Jacopo Mokachechi, we were part of writing up the deciduous form of the dental paper. So this is my mandibular P3s. When I looked at the overall shape of those teeth, again, the lower third premolars, they look very similar to each other. And I already told you they have a unique morphology that doesn't overlap with any other hominids that I have been able to find. But also that they're so similar to each other. So we say they have low intra-specific variation. And that's pretty amazing. So we look at teeth such as from Australopithecus africanus, like Mrs. Plez is, some other Australopithecine teeth, there's lots of variation to their shape and size. And we don't see it with Homo naledi. This is one of the lines of evidence that helps support or suggest that these individuals might be related to each other. Again, they look very similar to each other. The other thing is that I looked at, other than the overall crown shape, is look at some of the relative cusp analysis. So your upper molars tend to have 4 cusps, your lower molars tend to have 5. And so we look at the size and the shape, the proportion that cusp makes up of the tooth and how that shifts over time. So that's something else that I've been working on. And the adult teeth, but again, I've been focusing a little bit more on the deciduous teeth of late. So as we continue down, just quickly, the rest of the body is that we look at the shoulder area. And so we have the shoulder blade, part of this here. And from that, I'm sorry, going the wrong direction here. From that, you can see the glenoid socket up here. This area part of the scapula or the shoulder blade is angled up. And so that tells us that their shoulders were up a little bit higher than ours were. And this would suggest a little bit more shoulder, a little bit more mobility in their shoulder girdle. Here's, we
Juliet?
have, yes?
Before you get too far from the teeth, a question about cavities. Did I see cavities there, or was that just where?
Just where we don't have any cavities.
Okay.
Yeah. Okay, so then for the rest of the body, with the thorax, it looks like with some of the ribs that we have, it looks like it gets more narrow towards the top. That's definitely more primitive, more ape-like condition. We have small vertebral bodies. So some of these things are also suggesting, again, a lot more mobility up at the shoulder girdle, suggesting that they might have spent more time in the trees, or at least had the ability to spend time within the trees. The muscle markings on the arms, we have parts of the humerus as well as the some of the forearm bones, the ulna, the radius. And so from those, we can also reconstruct that they would have had the muscle markings, suggesting that they would have spent some time, or again, at least could have spent some time in the trees. The hands, further evidence. So we have lots and lots of hands, not every single piece of the hand and wrist, but almost every other piece. So we had Tracy Kibble lead this part up and again, do a lot of great analysis on this. So from there, we can see the phalanges, the fingers are a little bit more curved. And so this is something else that helps support spending time in the trees, but we also have a human-like finger to thumb ratio. So again, that's something very modern. So this mix, even just in the hand. So here we have this. And so, I mean, we keep our hands super straight. We can curve them. These would not have been able to be a perfectly straight hand. It's also quite small. Here's compared to my hand, and I'm not a giant, so it's not very big. So yeah, there's a big mix of primitive and derived traits, primitive or ancestral, and derived or more modern-like traits that exist within the hand. So to me, another very fascinating part of this species is the pelvis. So we have some parts of the pelvis that suggest it was really wide flaring. So it looks very much like Lucy, like an Australopithecus afarensis that's found in East Africa, dates to around 3 million years ago, that typically have this very wide flaring pelvis. It's also compressed more front to back than what a modern human would be. Ours is more bowl shaped, not quite as wide flaring as we see with the Australopithecines, and also more rounded in the birth canal. And so again, this is something, a very, very primitive condition, nothing that we would have ever expected to be with a specimen that has this more modern like teeth, and again, the shape of the skull, but perhaps with the smaller brain size, it might be more correlated. So again, that to me is a very fascinating specimen. Okay, so how bipedal were they? Since they have all these adaptations for the trees, well, looking at the lower limbs, this is the femur, your thigh bone. We look at the angle here of the femoral neck. So there's nothing, this femoral angle here, that suggests it's very, very bipedal. And also the muscle markings on the shafts of the lower limbs also suggest, again, a definite biped. So here I put a femur of a modern human next to a chimpanzee, just showing the angle that a modern human femur is going to make. Again, your upper leg bone. And so you can see this is different views of that same piece that suggests, again, it's a very modern, like definitely an obligate biped mover. We also know that's not a fluke, because it's not just a unique, maybe just that one fossil, that one specimen looks like that, because we have lots of repeated elements of the lower limbs. There's long, slender bones, again, definitely suggestive of being migratory and great bipeds. Here's another example, again, of that angle of the lower femur, lower thigh bone, again, suggesting that what some know as the valgus angle, but that angle is showing that as we are a biped, your legs are going to angle in underneath where your knees are going to be. We also have a knee area right from the patella that's going to look very much like a modern human, very derived. There's a picture of the tibia or the lower limb bone. Again, very, very bipedal, long, slender limbs. So we have almost a complete foot. Again, not 100%, but we have some great characteristics to this. And we had a podiatrist, and a man that used to be a podiatrist that now is switched to paleontology and paleoanthropology, and Bernard Zipfel. And he said that if this person had walked into his office, he would not have bedded an island. It was a little bit small, and he said a little bit curved, so the phalanges are a little bit more curved. showing it has the transverse angle, but a little more exaggerated, but otherwise he would not have had an eyelid. So here we're looking at a little bit smaller of an individual, reconstructing what that foot is. But again, very, very modern, despite the fact that it has a little bit more curved phalanges. So again, fully supporting an obligate biped. So I really like this picture. National Geographic put it in, but it just highlights the Homo or more derived, more like us features versus the Australopith. And again, highlights Australopith or ancestral-like characteristics. But again, highlights this huge mix that we have of the traits. So again, human-like shape of the skull, but it's really a small brain size, primitive shoulders, as well as curved fingers, suggesting the time spend time in the trees. But again, a really flared pelvis. The hands are going to be both derived as well as ancestral. Again, the palms, the wrists, but the human-like thumb to finger ratio are very, very modern-like. So again, we have this good mix of characteristics, which eventually led us to name the new species from the dentilated chamber. So let's talk a little bit about the taphonomy. So taphonomy is looking at the bones themselves. So what happened to the bones after an organism died? So we know it has some small snail and beetle modification. There are 2 antemortem fractures. So these are fractures that occurred before death and nothing that occurred after the individuals got into the cave. And the third thing is that the old individual has very mild normal arthritis. So when you have arthritis, the cartilage between the bones starts to wear away, they rub on each other, they get a little shiny. So you can see what that would look like from the skeletal collection. Okay, not super interesting. It's more interesting to talk about what the bones don't have. They have no evidence of diseases or disorders. So we don't really know exactly how they died or why they died. We'll say that. We have no evidence of carnivore or scavenger modification. In most of the other cave sites, we have carnivores have dragged them in, or porcupines with very specific tooth marks on the bones, and we don't have any evidence of that. We also have no evidence of any water transport. So via either the sediment or the materials that have been in. Also, the context of the cave would also make this very difficult. I'll show you this here in just a second, but we don't have any evidence of water transport. So we combine that data with the fact that the collection is almost entirely made up of hominids. So if there was going to be a carnivore scavenger that brought those bones into the cave, they wouldn't selectively choose only Homo naledi to bring into the cave. Also, the same with water transport. They wouldn't selectively only bring hominids into the cave. And so we rejected several hypotheses and came up with the hypothesis that they were deliberately deposited. They were brought back by other Homo naledi and deliberately placed or buried in the cave, in that dentilated chamber. So in this section back here, the far back section, I think you can see my cursor. And so this is a very hard place to reach. There's one section called Superman's Crawl. Literally, they have to turn their heads, the cavers, and fly through like Superman to get through it. Go up this area called Dragon's Back, where you have these large pointy rocks that they climb up there. And then the bodies would have been down in this dentilated chamber. We think they actually got up to the top and probably dropped most of them into that cave, that section, and then would have come back out. The Superman's Crawl might not have geologically been exactly the same, but Dragon's Back looks like it would have always been then that kind of treacherous climb up into the dentilated chamber. So again, this is something that was a little controversial when it came out, as we rejected these hypotheses. The other thing is that this is deep in an area known as the dark zone. And so how did they get in there without fire? We currently have no data, no evidence suggesting they had fired, but we are continuing to look.
Juliet?
Yes?
I don't think you, or I may have missed it, to talk about the age of the site yet.
I have not yet.
Okay. Yeah, I'm just wondering if this is the earliest sort of record of caving, right? So people going into a cave on purpose, they clearly have already explored it before they would have dropped people in there.
Well, that's the next point. Yep.
Yeah. Okay. Well, fantastic.
But they definitely would have brought. I mean, somebody went in there, whether they had fire or not, and explored and found this area, right?
Yeah.
And the parts that I've been into, and these other South African caves I've been to, I mean, you put your hand in front of your face, it's as black as dark as night, right? I mean, you can't see anything. If you didn't know that was your own hand, you wouldn't know it was there. And so, yes, somebody would have had to explore this already. And so that to me is already fascinating. It's also a lot of effort to go and place your individual in this really hard to reach chamber. But again, they did it. And let me remind you, we have 15 individuals. We also know that the individuals weren't deposited all at once. It was already over a period of time that they brought individuals back into Dee Naledi. So that's site 101. 2 years later, in 2017, we came up with site 102. This is the Lassetti chamber. And so 101 is down here in yellow. Lassetti 102 is up here in red. It's a slightly different passageway, but there we have an adult male that looks like he's laid out on this edge. So we have most of his skeleton. We have part of a female skeleton and then a juvenile. They're somewhat spaced out a little bit, but again, that adult male is largely articulated in on the one area. So we've got 3 specimens that were found in here, and they have very similar or consistent morphology with the Dee Naledi chamber. But 2017 is also where we finally came out with the date. We had not had a date with the whole workshop. We didn't have a date, which originally was very frustrating because, well, that would help us to narrow down what it was, but it also helped us to think outside the box because we weren't limited by, oh, okay, well, it must be Homo because it's super young, or it must not be Homo if it's 3 million years old. So these different characteristics. So I think it was a very healthy exercise that Lee made us go through. That was, again, a little bit frustrating in the beginning, but we did not come up with a date until 2017 is when it finally came out. We had 5 different dating techniques from 10 different labs. So we're pretty secure about a date between 335 and 236. We had to be very secure about that date because, first of all, it's a very small brain size. And to have this modern-like shape, we would not necessarily, when I first gave talks on this specimens, I said they look roughly to be 2 million years old with this super primitive pelvis and this super small brain size. Maybe early Homo, basal Homo, not entirely sure, but 300,000 was very different than any of us were expecting. And so that leads to changes in some of what I talk about, some of the implications about reassessing what it means to be in the genus Homo. So this was site 102, super exciting. Last week, we launched a new paper, site 110. And so it doesn't have a fancy cave name, but it's still, or at least not anymore, none that we published on. But site 110, so we look at the map here, the new and updated, this is 110 over off to the side here. There was a paper put out by Marina Elliott that talks about the new cave explorations and all the different areas of the cave that we have found. Some have lots of specimens, some have just a couple kind of broken up specimens. 110 had kind of the most specimens. And so I went down and worked on those in 2019. And here we have these, so we have 6 teeth and 28 cranial fragments that came out of there. We have 2 deciduous or baby teeth, so the young adult teeth, the first set that you get that are worn. And then we have 4 adult teeth that are unworn. So we have them, they don't have any roots on them, they're still developing. So, and they're all directly associated with each other in this very, very hard to reach chamber. So hard to reach that the cavers, the pictures that we have, they're excavating upside down, reaching into this very hard passageway. We currently only have, because of that, we currently only have the surface finds from that. But we know that they're all associated with each other, the 6 teeth and the 28 cranial fragments that we have. So here's just one of the figures from our paper where we've taken part of the most diagnostic cranial piece is the frontal bone or part of the forehead, and then mirror image that. And then we've got pieces from the back of the skull. And we use that to kind of reconstruct an idea of what this early individual would look like. Some of these teeth up here are, again, because they're adult teeth that have not come into eruption yet. And so that's why you see some of those. I get lots of people ask me that question. Why are the teeth up in the skull? And so, again, super exciting that we have now a 3rd locality that we have hominid teeth found at. Number 2, we've got a number of the non-adults of the juveniles. We now have 5 juveniles. We've got 28 deciduous teeth, which is a lot to have for a hominid site. We don't have any other site that has that many. And we have, with our 5 individuals and a minimum number of individuals of 5, we have more specimens per individual. If that makes sense. It's not just like 1 tooth for each of the 5 individuals. So we can start to talk a little bit more about what it means to be a juvenile and a Homo Naledi. With our reconstruction here, with some of the pieces that we have, it's the first time that we have deciduous teeth in direct association with the cranial pieces. And so again, we can put that together. We can start to learn a lot more about growth and development with that overall association. And that's why it was such a big deal. Again, a third locality. And then lastly, the enigmatic behavior about what it means that there's a single skull, or even just if the whole child there, if that's in the sediment that we have not dug it up yet, that you've got a single individual of that's somewhere between 4 and 6 years of age that was placed in this very hard to reach chamber. What does that mean? 15 individuals in the first one, and again, 3 in the second one, and a single subadult in this third chamber. So that's some kind of the big takeaways from Site 110, from the newest paper that came out. From all this data, obviously there's lots of questions about the behavior, about what it means to be in the genus Homo, but I would say that the biggest question that I get is, where does Homo naledi fit in our evolutionary tree? And this is one suggestion that John Hawks came up with, but I mean, the short answer is we don't know. We really don't know. With this mix of primitive and derived traits, it's nothing that we would have predicted, right? And again, particularly with the very primitive pelvis and the small brain size, but with several of the derived characteristics that we find with the specimen. So, and then add to that the behavioral aspects that wouldn't necessarily be expected, but not completely unexpected for a species with such a small brain size. So I will say that we are definitely continuing to learn and need to continue to learn a lot more about Homo naledi. Thanks. I'd just like to say thank you very much to Blaine Schubert, Paleo Talks, for the invitation to present. I was really excited, and I'm really happy to know about them now because I've watched several of them already, and look forward to doing them more. And again, I can't thank enough the Rising Star research team, Lee Berger, everybody from that tooth booth. I worked a lot with, again, Luke Delazine, Matt Skinner. Again, we were on the descriptions, the analyzing, putting together this whole, putting together the idea of what all the teeth look like. And then, but everybody else on the project all contributed to this talk. And then Bernard Zippel and Cephalani for curators, and then some of the funding. So
this has just been, yeah, it's just been such a fantastic project to follow, with all the, the way it's been out there immediately and available to everybody immediately, and having such a team. It's just unparalleled, really. And I'm on the edge of my seat with questions here, and and having been there and having seen this landscape. And I remember, I think it was actually 2003 that when I visited, or it was 2002, I mean, that Lee asked me if I wanted to come down and start doing cave exploration, and I didn't, and I was just kicking myself. But the aspect of them being, taken back and dumped into that area, I admit it, originally I was like, oh, come on. And then you start looking at it, you start checking things off. What are the possibilities? Mm-hmm. You just don't come to any other kinds of conclusions.
People email me suggestions all the time, like
right.
A basilisk. Somebody said a basilisk brought them back there, like from Harry Potter.
Oh, that's right up there with a good option.
There was another one that the bones had literally been absorbed by the rock and reformed as they got into the chamber. Yeah. Oh, it's fascinating. My emails are fascinating.
I bet. I mean, the ritualistic aspect of all this, I think, is what makes it part of it so intriguing, at least, and makes us wonder about, are there any artifacts that are being found?
Right, not yet, but
yeah.
We continue to look. But it also doesn't seem, like a place that they're living.
Right.
So, it's a place. It seems like a, for lack of a better word, a little more sacred. They're bringing the bodies back there and, protecting them from things chewing on them, and again, like a burial. And so the living areas, right now we hypothesize they're not going to be found in that, but we're looking. It's a big cave system.
The other thing that comes to mind, and I don't know if it's a possibility, it's total speculation, is that, is part of it also sacrificial? Yeah.
It's true. We do have with the age range is not unlike a normal cemetery, with some of the old and young and not as many of the prime age adults, but yeah, really anything's possible.
Well, I'm a caver. I love going into caves and imagining the animals that once walked through and were in them, and now imagining these small hominins, going through and exploring and moving through it and having torches. It's just, it's mind blowing. And I know when I took anthropology classes, we had a very set way that we learned about the evolutionary tree. And the way that the South African record has just completely turned this upside down has been really exciting.
Great.
And you being part of that must be really exciting.
I absolutely love it. Every time I put something up or tweet, I always finish it with a hashtag love my job.
Yeah. Chris, you have a question?
Yeah, sure. I mean, that's kind of part of that's part of this paleoanthropology kind of story now. I mean, the field has always been at the edge, the bleeding edge of innovation, and at the bleeding edge of kind of trying new things. And then on top of that, you guys bring in a whole bunch of new people, new names, new ideas, put them all into a room for 6 weeks and say, what's going on? I mean, this is, besides setting the discoveries aside, which are phenomenal in and of themselves, but then are there kind of methods, techniques, approaches, new insights and perspectives that you would kind of point at this whole process of having kind of new ideas at the table and new people at the table? Can you point at maybe 1 or 2 or 3 of those things that have come out?
I would say, I mean, I'm trying to think. I mean, I know I particularly use elliptical Fourier analysis. You know, that's not a technique Tobias was using in the past, right? Or Raymond Dart. A lot of the CT scans that we're using to look at the internal structures. Matt Skinner does a lot with the EDJ, the enamel that covers the tooth, and the dentition on the inside. So looking at that shape, again, that's a much more modern type of research, looking at the internal structures. Also with the CT scans, it's easier to look at things like the root morphology without, with the tooth still in the mandible. And so I think those are some of the things. Again, Heather Garvin's reconstruction of the cranium, being able to mirror image that and then look at the mirror image that and then reconstruct what the cranium is based on that analysis. I think that was very forthcoming. Tracy Kibble does a lot of looking at with the next engine scanners and scanning them, and then she can compare them. They almost look like heat maps when you're comparing the different phalanges to each other in those papers. And so, yeah, I mean, it seemed, it was a mix of traditional as well as these more cutting edge things on there. That again, what led us to the early career scientists with recent data, recent PhDs to be on that project. So I went down there in South Africa and worked on Australopithecus sediba in 2010. And so that's where I really started to build up my database of South African hominin teeth in particular. And I had a smattering of pictures from Daryl, my advisor. But otherwise, the majority of that work was trying to test whether Australopithecus sediba should be an Australopithecus or a Homo, because remember, that was a big controversy in 2010. And so trying to see where the crown morphology of those teeth fell and whether it was closest to Homo or Australopith. And so, my conclusions, that was part of our science paper, was that it always fell consistently closer to an Australopith, but never within any of the normal ranges of variation for those. The other big thing that I didn't mention yet is that Homo in particular in South Africa is fragmentary, scrappy, and heavily debated. I just had my students make the STW 151 is a specimen from Sturkfontein that's got this mixed dentition of adult and deciduous baby teeth, and lots of teeth, we even have some, fragments, some mandibular and maxillary fragments, and we have no idea. We argue whether it's Homo or Australopith. To which, personal communication with Jacopo, one day, Jacopo Mogiceshi said, well, why didn't we just have the 2 genera to choose? Like, okay, we're throwing out a third one in there. I mean, we just don't know a lot of what Homo looks like in Southern Africa. Now we definitely have an idea of what Homo looks like with Homo naledi. Again, repeated elements and all is directly associated with each other. Kind of went on a tangent at the end there, but.
Well, I mean, another part of this too, and kind of what Chris was alluding to, is just the amazing nature of getting all of these people down there to work together. And I think we're really envious when we look at this as and as an example of what we would like to be able to do with different fossil sites. And so if you could just speak for a second to how much better that was perhaps than the individual studying it here and then later another individual and then another individual. It must have been amazing.
Personally, yeah, absolutely. And I also think it would be hard, difficult, impossible for you to go down and have the wealth of knowledge that all of us had as a single or a couple individuals.
Yeah.
The other thing that I think is also very different is when we went down to analyze these. I mean, we're there for the 6 weeks, intensely studying these fossils, versus you came in, you studied it for a couple weeks, you got pictures and some measurements, and then you go home, and then we email back and forth and we talk about it, and then but something came up in life, so it's put off for a little bit, and you're back. I mean, 6 weeks, we had rough drafts, ideas, very, very rough stuff by the end of the project. But the other thing I really liked, at the end of the night, we'd get together, almost nightly, and we'd have our computers out at, while we're having dinner, having a beer and chit-chatting, and, well, what about this specimen? Did they have that? And again, all this communication between the different groups. Well, the pelvis is definitely an Australopithecus. We have to put an Australopithecus based on the pelvis. We're like, whoa, the teeth say nothing. No. We had these great debates until we couldn't anymore. We had to go to bed and get up and do it all over again.
Was there anybody filming all of this sort of like as if for a documentary?
There was some photography, but no, not major. Yeah. I mean, in that regard, they somewhat discouraged us from talking to each other. Not discouraged us, but
sure.
You wanted to base your ideas on kind of your part of what you were looking at and not be biased by any of the other aspects. But there are other people that came out in the literature said, I'm pretty sure it's really just a small Homo erectus. Mm-hmm. Well, if we put it in as Homo erectus, I took out a picture that it looks very much like a specimen from Olduvai Gorge, OH9. The shape of the skull, but that skull is twice, the brain size is twice the size, and it's 1 million years old. Mm-hmm. So how do you riddle me that? Number one. Number two, we would just have to change the definition of what it means to be a Homo erectus. And it's already a bit of a mess in what we put in there. But the pelvis in particular, it's known for having a very derived pelvis, a very modern-like pelvis. And you cannot put that very flared Australopithecine-like pelvis into that definition.
Well, let's go over to David and see what kinds of questions are coming in. We're having so much fun. I'm losing track of time here.
Sure. We have a lot of questions coming in from our audience. So as a reminder, if you haven't asked your question yet, you can go ahead and put it in the comments of our Facebook video. Or if you can't leave a comment on Facebook, go over to the Gray Fossil site, Instagram or Twitter, and send us a question there. All right, we're going to get through as many of these questions as we can. We might not get to all of them as we're getting toward the end of our program. Let's start with this one from Donna. Donna asks, is there any evidence of an ancient sinkhole above the chamber, which might explain the lack of torches?
With no geological evidence of that yet. I rely mainly on those geologic papers. This is the second or third one that's come out, but there's no suggestion that there's any other way to get into the D. Naledi chamber as the way they suggested. There's ways of getting into the other parts of the Rising Star Cave system. We do have the site of Malapa is a vertical death trap that the fossils probably fell into with Australopithecus sediba, but those specimens have breakages on them suggestive of a fall, and we don't have any breakages on the bones of these suggestive of that same fall.
All right. Here's a question from Jenny, who asks, will there be isotope analysis done on the teeth to figure out the ratio of meats versus plants in the diet?
Great question. There are, and there will be those analyses. Some of them are ongoing. So far, the microware structure, you guys have heard about that with Peter Unger. He included Homo Naledi in his discussions there, but there will be more isotope studies being done to look at their diet.
Great. That's exciting to hear. Jennifer asks, are there plans to continue excavating in the D. Naledi chamber?
Yes, and they had a small skeleton crew, pun intended, working on site 105, and I am jonesing to get back. So I have plans. I'm on sabbatical in the spring to get back there, see what's come out. I know some teeth have come out, but there are. This was site 110. They're, I think, up to site 111 now. Again, some of them are very small, scrappy, not much to them, but they're all those sites. 105 seems to be the most productive right now, but absolutely. I don't know they're going to go back to the D. Naledi chamber. I mean, once you have 15, what else can we learn? We have repeated elements of multiple repeated elements of everything. So, it's kind of a point of return. And it's dangerous to go back there.
That ties into another question we have from Gus, who asks, how many other chambers are there still to explore?
I'm sure there's a lot, and some that we don't even know about if we can't slip through, literally through these really small areas. And also, again, safety is a big concern. So they're continuing to explore a chance they can to really map out that whole system and make sure that we've seen as many as we can. So they continue to go down.
And I'll actually follow that up, since that's a really interesting line of discussion, that there is a balance, and this is the case with any fossil site, but especially here, since safety is such an issue, that there is a balance between it being worth the time and effort of going down there compared to what you're actually finding, compared to keeping everyone safe. At what point does everyone sort of agree that we've gotten everything we can out of this particular site?
Mm-hmm. I mean, I'm guessing that, is that rhetorical?
No. I'm actually asking, do you have a sense of, like,
I think
at what point does the safety issues and the amount of findings,
so
we just move on?
I think once, not yet. And we're certainly going to look at some of the easier to get places first. I think that there are some passages I'm not even sure we can still get to anymore. But yeah, I don't think we're far from that. Like I said, I think we're already there with Naledi. There's not much more we can learn from there. I am a little bit more interested, am I being honest, I'm more interested to see what would come out of the rest of 110. But it's either that they just put a skull, or they dug a hole and placed a 6-year-old in it with just the skull exposed. Either way, it's weird behavior. It's interesting behavior. But so I'm interested in what might be in the soil below it. Again, would that really change anything? So yeah, I think we're going to continue to map out the cave, get a good idea of where everything is, and then slow down. We're also trying to, there's some areas that we brought up, massive blocks of fossils of the sediment that have complete dentitions in them, because we don't want to lose the provenience. Once you open that up, they fall apart. So we've CT scanned those and are trying to pull out individuals, and we'll probably just print those. So I think actually that's another more modern aspect, things that we've done with the site, because once you again open that up, we're going to lose the position. And it looks like, I've been counting them on the CT scan. It looks like there's an individual right above the dentition, right above the permanent, and then the juvenile is right below it. So there's just a lot of teeth right on top of each other versus multiple individuals. So I don't know the answer to the question, but I don't think we're far off from it.
Gotcha. Well, thank you for giving your insights on that. And speaking of the sort of pattern of excavation, here's a follow-up question from Jennifer, who asked, is further excavation or analysis continuing to be done by early career researchers in a similar team style? I remember how exciting it was to follow the news as it started to come out, and how it was such a unique opportunity.
I would say it's not. Yes, I would say it's continuing to work that way, but maybe not quite as strong. I mean, there's not as many primary analysts. I mean, I had my student working on it. She did some analysis in 2019, last time we could go. I think it will pick up again once COVID changes things and we can get back to exploring. I would say personally with my grad students, we have an idea of what you want to do for your projects, but also the COVID contingency plan. If we can't get anywhere, what could we do? So that would change. You wouldn't have as many early career scientists working on these specimens the way that we had in the past, again, just for mobility purposes. So I think it will pick up again once the world opens up a little more.
Okay, and I'm going to go ahead and ask one more, just to make sure we have time in case Blaine or Chris have anything else they want to say. This one's from Benjamin, who asks, as a lumper, how do you feel about identifying a new species of hominin? Does this change your views on early hominin diversity?
This has changed my, I also liken myself to be more of a lumper than a splitter. And I just don't see how I can lump this in with anybody else. The big question is, I left up some slides in case anyone asked about other things, but is the coexistence. We now know that we've got anatomic and modern Homo sapiens in Morocco, a specimen called Jebel or Hood that dates to 300,000 years ago. So this is the first time we have two species coexisting in Africa at that same time, two species of Homo. And what does that mean? Now, Morocco and South Africa are very far apart from each other in Africa on this massive continent. We don't have any data that they were right, that they had a direct association with each other, but what would that mean? Would they have interbred? Could they have interbred? I mean, I don't think it's outside the realm of possibility, but I don't know. I lean more towards lumping, and there's nothing in the specimen that suggests to me that it would not have been, that it isn't a separate species.
And do we think that, I mean, this group is branching off and relatively isolated, perhaps, for it to retain certain characteristics like the pelvis? That has, when you're combining these primitive characteristics, while on the other hand, you have Homo habilis that is moving in a different direction, and then this one is lasting longer. Is this some kind of an isolation, or?
That's what I think. Yeah, more of a genetic drift
Yeah.
characteristic. Yeah, in South Africa. So they probably didn't have direct interaction with modern humans for very, they probably were constricted from it for quite a while.
Fascinating. Well, we better wrap it up there. We're at time, and this has just been so exciting. Thank you for joining us, Juliet Brophy.
Thank you very much for having me.
Wonderful. All right. We'll see everybody next time.
Cheers.